Thank you, Paul, for the invitation, and thank you everyone that is here and those that are online. So in the next few minutes, I'm going to be discussing on the potential application of a DNA methylation array in cattle, using the Canadian dairy industry as an example. Now, from the previous talk, we saw the application of DNA methylation array in the humans. So we hope that in livestock research, we'll get to that point one day. So my talk will be in three parts. Firstly, I'll give you a brief introduction on the Canadian dairy industry, some brief statistics, and then some indicators from my laboratory, and then we go to talk about the hypothesis, the potentials. So before I get to that, just want to we'll give you a brief presentation on my research center, which is from the Shelby Research and Development Center, which is one of 20 research centers of Agriculture and Agri-Food Canada. So you are familiar with USDA, INRI, and all that. So I don't know if there's similar structures here in the UK. So Sherbrooke, my center is the blue one here, and it's one of 20. So all this city, there are various different centers, and every center has a different concentration. My center is concentrated on dairy and swine research. And my little city is a beautiful one that is surrounded by nature and water, and is 180 kilometers east of Montreal. So if I say Sherbrooke, you'll be hard to place that, but if I call Montreal, you can easily place that, and it's also close to Boston, the US, and New York. Those are the closest big cities. So my center, the main mission is to provide dairy and swine producers with technological innovations, know-how, so they can profitably produce high-quality milk and products while ensuring animal health as well as the health of their environment. So every 10 years, our research focus must tie in with the strategic plan of science for the Department of Agriculture and Agri-Food Canada. And starting this year for the next 10 years, there are 4 missions: mitigating, adapting to climate change, advancing the circular economy, and increasing resilience of agro ecosystems, and accelerating digital transformation. So our farm, this is from the sky, that is how it looks, and it is on 400 hectares of land. We have the dairy complex, swine complex, environment complex, farm buildings, farmlands, and where we have our offices and all that. So these are the faces behind the talented people I work with, different groups, but all working together as those are the various expertise. And we work in collaboration with universities, other federal and state, provincial labs, universities, research organizations, and international partners. And importantly, we have at any time T1 about 100 graduate students working in our laboratories, as well as exchange students and interns from all over the world that come and acquire research experience working on our projects. Now, going back to the main focus of today, the brief statistics on the Canadian dairy industry. Dairy farming is very, very important. So Canada is the second most important activity in the agricultural sector. In 2019, it provided a total of $20 billion to the Canadian GDP. And in terms of jobs, for a country that has 39 million people, the dairy industry provides jobs to about 2 million people. So, and the products, they're high quality milk and dairy products for domestic as well as for exports. So today, the number of dairy cows in Canada, we consider the cows and heifers, is 1.4 million, and farms is under 10,000, and the milk production is about 92.4 million hectoliters. Organic milk production is getting more and more importance because of consumer preferences, and goat milk production is also catching up to the European counterparts. So we'll look at the provincial distribution. My center is located in Quebec because it is the most important in terms of dairy production in Canada, having the most farms and the most cows, followed by Ontario. The other provinces are mainly concentrated on cereal and oil crop production. So the reason why our center is located in Sherbrooke. So The dairy cows, when we look at individual breeds, the main breeds that contribute to our dairy production is the Holstein, constituting 93% of the national herd. So Jersey follows at 4% and the Ayrshire at 2%. The other breeds, like the Canadian and the Milking Shorthorn, they are quite few in numbers, but they are well maintained and kept for the sake of genetic diversity and a store for future use. Okay. Now, if we look at this graph here, it shows the dairy production in Canada, the output with the introduction of genomic selection. And genomic selection was introduced in 2010, from 2010, and we see that the milk production increased rapidly, even the protein and milk fat percentage. So from 2011 to 2020, due to genomic selection, the milk production increased 2.2-fold increase. Now, if you look at the evolution starting from 1970, we see that the Holstein cow produced about 5,700 kilograms of milk per lactation. And 10 years later, it increased by 1,000. And 50 years later, that number was 11. That number was 2021, 11,094 kilograms. That is doubled. And we see a similar trend for fat percentage as well as for protein percent. So if we look at these numbers, we see that the annual milk production per the most important breed in the dairy herd doubled in the last 50 years. And what could have been responsible for these technologies enhanced the livestock breeding. Now, starting with improved management practices, nutrition, improved healthcare, housing. And when we talk of improved management on Canadian dairy farms, we have what we call the pro-action plan that has been instituted by Dairy Farmers of Canada. And all herds, all farms that participate in the Dairy Herd Improvement Program, they have to follow a certain rules, right? And regulations, rules, protocols that they have to follow at the farm to make sure that they produce milk of high quality, ensure the safety of milk, as well as animal care, traceability, biosecurity, environment, and all that. So for that reason, the milk that comes from the farm is of high quality. And this is also seen with the somatic cell count on Canadian dairy herds that is averagely at 200,000 cells per mil. Now, if we look at other technologies, from when performance recording was instituted, and then with the advent of artificial insemination and embryo transfer, we saw the impact on livestock production throughout the world. And then genomic selection, the previous graph I just showed, resulted to a twofold increase only over a period of 10 years of introducing genomic selection in Canada. Now, what about the future? What about future technologies? We are here today talking about epigenetics and what it could do to the industry. So let's assume hypothetically that epigenetics could be one of those technologies. Let's go ahead and see what happened. Now, the genomic breeding, how did that happen? It happened because of the use of genomic information aided by the availability of the genotyping chips that was used to genotype those bulls and cows and implemented in breeding. And we saw the result, which are higher rate of genetic gain, increased reliability of predicting breeding values, shortened generation interval. The bulls can be selected at a very young age, as we just saw with the previous presentation. And with that, there's rapid genetic improvement in all traits, including very lowly heritable traits like fertility, lifespan, and health traits. Now, if we look at this graph here, we can see the 5-year genetic gain that was realized on Canadian Holsteins before genomics was just like for milk yield, that's a highly heritable trait, about 345 points. But with genomic selection over a 5-year period, that number almost like doubled. And now when we look at traits like mastitis resistance, the response was like low, but with genomic selection, that also doubled. And traits like lactation persistency that responded negatively to selection. But with the introduction of genomic selection, those traits, it became now positive. Now improvements start to be realized in those traits. And here in this graph here, it shows the amount of what was realized in terms of the lifespan profitability index. Now it's an index that is done. If you want details on that, you have to look at LactaNet website. So the gains that were realized for the lifespan profitability index for each cow annually before genomics introduction was just 45 points, but with genomic, that will double to 97 points per year, as well as there's this index is called the annual pro gain, which also before genomics was just about $102 gain per year, but with genomics, it now went up to $246 a year. That is, those are huge gains for the industry. But with all those gains and with all the positives that we realize with genomic selection, but yet we cannot just stop there because there's still increased demand for animal products. And with the impact of animal production, especially ruminant production on the environment, it is but logical that we concentrate and keep fewer animals that produce more products than keep too many animals, knowing the effect that they have on the environment. And also the information we use for genomic selection, well, we all know that they do not sufficiently explain the phenotypic variation that we observe. And so moving from the phenotype, the component, we see now that there is some missing information, and our various experiments in our labs, looking at epigenome, we all find that they affect traits. So they could be part of that information that could be quantified and used to further realize gains in the phenotype for phenotypic expression. So our business here in these 2 days is to understand, to find ways to capture the epigenome influences efficiently and cheaply and exploit such and its use in genomic breeding. So with that, I'll give some brief indicators from some research work from my lab. Now, I just want to talk briefly on a study that we conducted, trying to look at the DNA methylation alterations between cows producing very high milk yield or very low milk yield, very high protein percentage or low protein percentage. And we characterize the mammary gland tissues from these cows by whole genome by sulfide sequencing. And we use a few cows, right? Because the method does not permit cost-wise to use too many animals. So when we found the differentially methylated cytosine, and on this Manhattan plot, we found some regions, these regions that are highly supported here. And you know, when you do your GWAS analysis and you find such regions, you're very happy on that day that you even have to go out to get a beer. So when we found this, it was very interesting to us. So we zoom into those regions to see what was happening after we perform co-location analysis with reported QTLs for those traits, for milk trait and milk composition trait. And we found that this region here on chromosome 27, we can see these DMCs here that are highly supported. And at this region, there are many QTL that are located at this region. And also here for this region on chromosome 6, we also found many QTLs that are located here. So with a few cows, we cannot say much about this. So we hope that with an epi chip, we can have more information. So here was another study that we performed to characterize the DNA methylation alteration in ileum and ileum lymph node tissues from cows with subclinical paratuberculosis infection. So the ileum and ileum lymph node, we know they are all at the same place, at the same location. But when we did the characterization after whole genome by surface sequences, we found high numbers of DMCs, differential methylated regions, that were differentially expressed between positive cows and negative cows. But overall, the result shows that the ileum and ileum lymph node, they responded differently to the effects of DNA methylation, where the ileum lymph node was more responsive to DNA methylation. That is, by more alteration, we found more sites that were altered in the ileum lymph node than in the ileum. And so this being tissues at the same location is an example of the tissue-specific expression that we associate to DNA methylation alteration. Here is another study, a recent study that is still ongoing. And so we characterize milk somatic cells from cows that are positive to Staphylococcus aureus mastitis or Staphylococcus chromogenes and versus healthy cows. So we characterize both the methylome, the whole methylome, using whole genome methylation sequencing, the enzymatic method, and as well as the transcriptome. And so when we integrated the result of the methylome and the transcriptome, we found the inverse correlation between DNA methylation at the promoter regions and the gene expression, as well as at the first exons and gene expression, as well as the first introns and gene expression. So as the methylation level decreases, the gene expression level increases. But when we look at the inner exons, we didn't find this relationship, which was okay. The first three, the promoter, first exon, first introns. So this relationship was expected, and we're happy that we saw it this way, because already in the literature is reported like that, and these are considered the regulatory regions. So we've based our further analysis on these regions. So globally, the methylation alteration in the Staphylococcus aureus samples were higher than the control samples, as well as in the S chromogenes was also higher than the control samples. So here is just a summary of the differential methylation sites between positive sample and control, and this positive sample and control at the level of single CPGs, CSGs, CHH, differential methylated regions, and methylation haplotype blocks. But what was very interesting for us is this column, whereby we found so many sites that were common between the two pathogens. So hopefully in the future, when we are able to use more samples, we'll be able to actually find those that are specific to each pathogen. And we went ahead to look at we used the program Diablo from Mix Omics, and we're able to find some DNA methylation haplotype blocks, as well as some few genes that were able to explain most of the variations between positive samples and negative samples. So, and this is what I can say the first step of identify some markers. So we hope that with further experiments, we'll be able to validate these markers, that actually they can separate the sick cows from the healthy cows. So I want to talk a little bit about the various methods that we use, the characterization as single CPG sites, which is what is easily done, that we all do easily, but we all know that it can be influenced by technical factors such as batch effects of samples, as well as the sequencing depth. We also look at differential methylated regions, which we can consider that more robust because it helps us to reduce the large amount of data to just a few sites that we can concentrate on. But to identify them, we have to define certain windows along the genome, so through that we can miss some site, and also some site could be very long, like in a 1 KB region, you can have a DMR there with just 3 CPG sites, so which make it difficult for downstream analysis. So the other strategy we use in our study is the differentially methylated haplotype blocks, which is a block of tightly coupled CPG methylation site. So methylation site that co-methylated adjacent and coupled CPG site. So the methylation haplotype blocks, they are shorter inside, so make it easy for downstream study like targeted by surface amplicon sequencing, or can be helped in the selection of sites with epi chip, because we can easily prioritize some sites. So here is an example of a methylation haplotype block that we identified. So this one is on chromosome 1. The length is 105 base pairs, and it had 10 CPG sites. And we can see here that in the Staphylococcus aureus sample, it is hypermethylated, but not so in the control samples. So here we can, in the case of downstream study, you want to identify, you study one site, you can just select one from this block because they are tightly coupled together. Now we went ahead to look at how those samples, the methylation haplotype blocks, we went ahead to validate them in a higher number of samples. Initially, our ambition was to select like 200 important sites and validate them targeted by surface amplicon sequencing in over 1,000 samples, which we have, but we could not do that because of the cost associated with that. So we limited our samples to 100 samples and 9 sites, which costed us about $21,000 to do that. So we selected 100 cows with high somatic cell count and 100 cows with low somatic cell count for this study. So after our amplicon sequencing and data analysis, this PCA plot here tended to divide our samples that we selected as samples with high somatic cell count into 2 groups, which was surprising, but again, not surprising to us because our only criteria was somatic cell count. So we didn't have further information whether those samples there was a pathogen or the type of pathogen in it. So it was surprising, but not surprising. And the control samples on this side. But what was surprising to us, because one of the samples in the high somatic cell count group, they had similar status, methylation status as cows with subclinical mastitis. Now, when we look at association analysis between those methylation haplotype blocks, they were on 2 genes, on the promoter of 2 genes, the AVPL genes and the MF2A gene. And we can see that they were significantly associated with somatic cell count and somatic cell score, as well as with milk production, either with high milk production or low milk production status. Now, our next steps is to use more samples. If Helen succeeds with the epi chip, we'll be able to do that, to use that on our samples that we have, and also to fine-tune our classification of animal. We have more information on the health status. Now, that said, we go now on our hypothetical, trying to see what the availability of a DNA methylation haplotype epi chip can mean to the livestock industry. We all know that the current methods that we are using now, they are NGS-based, they are costly, even though they are good, they give us good resolution, but they are relatively cost prohibitive, and we can only do that on a few samples. Meanwhile, our traits that we focus on, they are quantitative traits, needing that we have to have information on a large number of samples. So from what we learned yesterday and what we know from the literature and what was presented earlier with the human chips, DNA methylation chips from Illumina, we know the workflow is simplified, high reproducibility, cheaper, and time and also cost effective and can enable a wide range of studies. So we hope to have that soon. So with that, for sure, it will enable studies like epigenome-wide association studies for a wide number of traits, and with that, it can help us to identify candidate markers, causal epigenetic markers, like the last presentation, that we can have such information from a large number of animals. It can help us in health to manage the health of the animals, like it can have diagnostic value, prognostic value, predictive therapeutic value, as well as for monitoring. Now, if you look at disease diagnosis and risk assessment on the farm, it can help us to separate high-risk animals from low-risk animals, as well as identify cows that have chronic conditions like mastitis or subclinical mastitis, and so on. So what is happening in humans now, there's already in the era of producing epigenetic drugs, epi vaccine, and so on. So the applications are limitless. So in terms of managing livestock with the epi chip, it will help us, it will provide us more insight into the interaction of DNA methylation alterations with other organismal processes, and like at the biological level, the molecular, cellular, and immune level, and will help to provide insight on how to shape the phenotype outcome and how it can be exploited for livestock management. So, as I already said, the applications are limitless. So, the epigenetics, its availability could further help us to improve and enhance the genetic gains in current livestock breeding. So I presented this graph earlier and how it has helped to further gains in the Canadian dairy industry. So could the epi chip help to take epigenetics information to the level of a future technology? And if that happens, and if the low density and high density chips are available, and that the industry starts to use that to enhance to use that for breeding and implement that in breeding. And we can say that we are today in 2023. By 2028, can these numbers, can they go up like this? If that happens, the farmers will be very happy. So in conclusion, if an epi chip is available, it will help us to further understand epigenetic basis of livestock traits, and it used to achieve further gains in dairy health management and in production. So with that, I would like to thank my research team, especially the graduate students that did most of this work. Menchi Wang, they did most of the work. So I want to thank them, our bioinformatics group, as well as the participating farms and Agriculture and Agri-Food Canada for funding our research. And I would like to thank you for listening. And if you have any question, I'll be happy to answer. Thank you.
Thank you, Eveline, for a very good presentation. I think maybe just take you back to your slide, looking at the projection of what is possible with application of epigenetic. So if you look at your equation there, so what really matters is how much we can improve genetic variance by including methylation, DNA methylation information. So basically, has any study been done to actually quantify the amount of additional variation that DNA methylation actually brings to the table? For instance, let's say take milk or fat. What additional variant does it explain in addition to what we already know? Is there any study in that direction?
All right. Thank you very much for that question. So why we are here is because from our various small studies on DNA methylation in livestock, right? The methods available can only go on a few samples. And we need the methods that we can use on a large number of samples. Like our study on mastitis, we were ambitious, we wanted to, after select a few markers and do an association analysis on 1,000 samples, and we actually have those samples. But the method, we cannot quantify that on a large number of samples. And you are the ones that does the equations and those calculations. You need the information on a high number of samples. And it's the main reason why we are here yesterday and today, looking for how we can develop a tool like that for bovine that can enable such studies and how that information can be captured. For now, no, there's none available.